cell biology, and genetics. The branch of biology which deals with the study of structure and functions of the cell is called cell biology. The branch of biology which deals with the study of heredity and variation is called genetics. Cell division. Cell division is a complex process by which new cells are formed from pre-cells. The cell divisions are of three types: mitosis, mitosis, and meiosis. Mitosis. This cell division is common in bacteria. It is a simple type of division where the cell contents, including nucleus, move into two by an inwardly growing constriction in the middle of the cell. Mitosis. Mitosis is a common type of cell division, usually occurring in body cells. In mitosis, the number of cells is multiplied without change in the number of chromosomes. It takes place in 5 phases. Phases of mitosis: prophase, metaphase, anaphase, telophase, cytokinesis. Meiosis. Meiosis is the type of cell division where chromosomal number is reduced to 1/2 in the daughter cells. It takes place usually during gamete formation. Stages of meiosis. Meiosis 1. Meiosis 2. Meiosis 1. Prophase 1. Metaphase 1. Anaphase 1. Telophase 1. Prophase 1. Leptotene, zygotene, pachytene, diplotene, diakinesis. Meiosis 2. Prophase 2. Metaphase 2. Anaphase 2. Telophase 2. Meiosis takes place in 2 major steps. They are meiosis 1 and meiosis 2. Meiosis 1. Meiosis 1 includes 4 phases, such as prophase 1, metaphase, anaphase 1, and telophase. Prophase 1. Prophase 1 is the longest and most complex stage. The sequence of events of prophase is divided into 5 stages, namely leptotene, zygotene, pachytene, diplotene, and diakinesis. Leptotene. The fibers of chromatic condense and become a thread-like structure. Hence, it is called chromosomes. At the end of leptotene, each chromosome consists of 2 chromatids. Zygotene. 2 identical chromosomes, in other words, homologous chromosomes, come together and lie side by side along their length. This is known as pairing or synapsis. Pachytene. The chromosomes condense further and become shorter and thicker. The segments of non-sister chromatids are exchanged in the region of chiasmata. This process is known as crossing over. Diplotene. The homologous chromosomes condense further and they begin to separate from each other except at chiasmata. Diakinesis. The chromosomes separate completely after crossing over. The nucleolus and nuclear membrane disappear. The formation of spindle fiber starts now. Metaphase 1. The spindle fibers become prominent. The paired chromosomes or bivalents align on the equatorial plane. Anaphase One. The two chromosomes of each bivalent separate from each other and move to opposite poles of the cell. Thus, only one chromosome of each homologous pair reaches each pole. Only half the numbers of chromosomes are received at each pole consequently. At this point, each chromosome has two chromatids, one in its original and another chromatid is a mixture of its original and the non-sister segment chromatic due to the crossing over. Telophase 1. Telophase 1 brings the first meiotic division to an end. Reorganization of the chromosomes at poles happens to form two haploid nuclei. One nucleolus may appear and the spindle fibers disappear. Meiosis 2. The second meiotic division is similar to mitosis. Meiosis 2 takes place in 4 phases such as prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Prophase 2. Nucleolus and nuclear membrane disappear and spindle fibers are formed at each pole. Metaphase 2. Chromosomes move to the center of the cell. They get attached to spindle fibers. Anaphase 2. The sister chromatids separate from one another. The separated chromatids are drawn to opposite poles due to contra of spindle fibers. Telophase 2. The chromosomes begin to uncoil and become thin. They reorganize into nuclei with the reappearance of nucleolus, nuclear membrane in each pole. At the end of the single cell will contain 4 haploid nuclei. Cytokinesis. Because of the formation of cell plate which becomes cell wall, little later across the cell is divided into 4 daughter cells. Each one is having haploid nucleus. Differences between mitosis and meiosis. Mitosis. Mitosis takes place in somatic cells for multiplication and to replace the worn-out cells. Meiosis. In meiosis, it takes place in reproductive cells, resulting in the formation of gametes. In mitosis, the chromosome numbers of the daughter cells are same as the mother cell. In meiosis, the chromosome number is reduced to half in the daughter cells. The prophase is a simple process of a short duration. The prophase 1 is complicated and of long duration. There is no pairing of chromosomes. Pairing, synapsis, takes place. No crossing over or chiasma formation occurs. Crossing over and chiasma formation occurs, causing exchange of genetic material.